Continuing chapter 11, we are going to do a whirlwind tour of the major phyla of animals, throwing in some terms that are going to be important as we look specifically at animal function. So we're going to work our way up from the more primitive, basically the oldest groups, up to the more modern groups, the groups that have evolved more lately. Primitive is a bad word to use because these animals have been around a lot longer than our modern groups and they have been very successful. So primitive somehow implies less than, and they are not. Now this group is very often considered a different kingdom. It's not clearly connected to the animal kingdom in terms of genetics. Kind of depends on what study you read. But these are the sponges. Three basic groups, whether their internal structure, reinforcement structure, or calcium, silica, these are the glass sponges, or protein. You don't need to know the specific names or actually those materials. But you do need to know that they're called porifera. And a few terms that apply to sponges will apply to other things as well. They, as a group, are not mobile. They are sessile. They sit in one place. Now, this can be applied to animals that are capable of moving, but only under duress, basically. The hydras that we looked at in the Nidaria lab, and I had out for the classification lab, they sit in one place and feed. If they get annoyed, they can actually do little somersaults and move, but they're still considered to be sessile. So sessile isn't incapable of movement. It's just they naturally sit in one place for very, very long periods of time. And they derive their food by straining water, by straining particles out of water. That's going to be their food. And this, of course, is something that happens in very small animals, and it happens in very large animals, is that the baleen whales, the biggest animals that have ever lived, are also filter feeders. It's just they got bigger filters, and the particles they catch are bigger particles. So this is a very common way in a number of different groups of getting nutrition. These guys have two cell layers. They have ectoderm and endoderm. There is a layer in between that is not considered to be a cell layer. It has cells in it, but it is mostly non-cellular matrix. These guys are not considered to have tissues, even though this is epithelium, and the jelly in between is basically a connective tissue. But at some point when people were doling out the definitions, these guys are not considered to have tissues or organs. Organs makes more sense, but tissues, I'm not too sure. But they don't have nerve tissue. They're not a coordinated animal. There's no connections other than chemical connections amongst them. And basically they, here we go. They have these little cells. And the cells have a little flagellum and around, not gonna show it too well in this. Let me throw a bigger picture of it up. This is in the endoderm. This, they call them, used to call them collar cells. Technically, they're choanocytes. And although this looks like a solid thing, it's actually a whole bunch of filaments, not microvilli, but filaments with little gaps in between them. And the flagellum pushes water down, and it goes out between the filaments and catches particles. And the membrane can take the stuck particles and move them down to the cell body where they're absorbed in food vacuoles and digested, and then those nutrients are shared with the non-feeding parts of the animal. So this is not colonial, but it's pretty close to a colonial organism. And actually there are sponges where if you were to put them in a blender and totally pull all of the cells apart, and then you poured them into the right kind of container, you'd get the sponge back. The cells would find each other and reform the sponge. What makes them not colonial is that if you separated them out, they wouldn't be able to live on their own. They can't do all of the functions that a living thing would need to do, but they can, disrupted, they can put themselves back together. And not all sponges can do that either, but it kind of shows you how much on the edge between single cell colonial and multi-celled you can be when you're looking at these earliest of animals. So, oops. Two cell layers plus what's called a mesoglia, a middle gel layer. These do not have an organized support system, but they do have internal structures that do support them. Those structures are called spicules. And the spicules are made of those three materials. Is that the calcareous sponges have calcium carbonate, similar to what we have in our bones. The silica, the glass sponges have silica, which is not just a great support medium, but basically like shards of glass. These things, almost nothing can eat them. And the protein-based sponges are what the original bath sponges were. They would harvest those, and those were the original sponges before they developed an artificial version of that. So that's the first of the phyla. Cnidaria, which used to be called Coelenterata, which is Latin for internal space. Nobody really liked that. Jellyfish with little outhouses with jellyfish on them, and on the caption it goes, only they know the difference. It does turn out that jellyfish, a lot of times are the sexual form in the alternation of generations. And telling a male from a female jellyfish just involves figuring out what cells they make. They look structurally pretty close to identical. Really is, only they can tell the difference. So, Cnidaria also has two cell layers in mesoglia. Oh, there was something I should have mentioned up here. I'm going to stick it up here. Trying to squeeze this stuff onto a video, sometimes I rush. Amoebocytes, sometimes called amoeboid cells, they crawl around. They are replacement cells, what we might call stem cells, where when cells are eroded off, or they get old and die, or they get sick and die, in both the sponges and the Cnidaria, these can move into the position that the dead cell or lost cell used to be in, and become that kind of cell. So they can form collar cells, they can form outer cells, they can form in Cnidaria stinging cells. And the idea that your cells need all of the genes because they need the capability of using different genes to be different types of cells, you can kind of trace back to these early forms. Keep in mind, that's not the only way to build an animal, is that there are some of the weirder small phyla that early on during development, only certain chromosomes go into some of the cells, and those cells are going to use the genes on those chromosomes to be what they're going to be, but they can't be the other type of cells because the genes for those other types of cells have gone a different way. And it is possible to build animals that way. It's just mostly every cell has the full complement of genes. Cnidaria also have the two cell layers. They are named for their stinging cells, what are called cnidocytes. And again, they're not considered to have tissues, even though they would be unhappy to know that. Sometimes they exist as colonies, where there's a whole bunch of connected individuals that are all doing, not necessarily all doing the same thing. The lab shows you a very simple colony with just two types of individuals in it. And depending on the type of cnidaria you're looking at, certain of the cnidaria that are sessile, like corals, movement is kind of useful for sexual reproduction, getting the sperm to where the eggs are. If you're stuck in one place, then you need to release the eggs into the environment and release the sperm, which gives you kind of a limited dispersal range. Sperm not going to be able to swim that far in order to get to the egg cells. And there are cnidaria that act that way. But a lot of the corals, the sessile coral is making more and more members of the colony asexually. So the whole colony is genetically the same. They can also bud off, right? Budding, one of the forms of asexual reproduction. Little mobile forms that are little bitty jellyfish. And those can move up into the planktonic layer, the layer that's drifting at the surface, and go long distances and find each other and mate, and then produce larvae that will drop to the bottom. Now, islands in the oceans, there's all sorts of volcanic activity and movement of the plates and stuff, and islands both sink and rise. And when the bottom of the ocean rises up far enough, it becomes a suitable habitat for corals. Only if corals can reach them. This can be a long way from the nearest coral reefs. But with these little jellyfish, medusae, spreading through the drifting water, they can colonize in a way that sponges cannot. Sponges don't have this kind of dispersal. The sessile form is called the polyp, and the little jellyfish form is called the medusa. Now, if you've ever been to the ocean and seen a jellyfish, it is technically a medusa, but it is not an alternation of generations medusa. The corals and a lot of their relatives, they produce very small medusae that are dispersal planktonic layer things. There's one unusual thing about this group is that some of the subgroups have taken one of these two forms and just settled on it and made it bigger. The anemones are large polyps. They do not go through a medusa form. They're these big barrel-shaped with tentacles on top. And of course, anything almost big enough to see that's a jellyfish is a jellyfish for its whole lifetime, except maybe it's a larval form, where they're not alternation of generation jellyfish. Now I'm going to show some pictures. I've been waiting until I got this off of here. So this is a polyp form. The hydra also has settled on a polyp form, probably, but even though they're quite small, probably because they have invaded the land, well, the fresh water outflows from the land, and flowing water and tiny jellyfish don't mix too well. Is that you're drifting, but you're drifting out to sea, so it's not a great dispersal form. So hydras can reproduce sexually. They can produce sperm and egg cells, be close to one another and reproduce that way. They can also bud off asexually and produce little genetic copies of themselves. This is showing the stinging cells. They come in a huge variety. This is just an example where There's a little trap door. There's basically chemical receptors on the top of that that can tell if a piece of sand bumps into it. They go, "Oh, okay, that's not worth worrying about." But something with protein, something that's like a living surface bumps into it, and they go, "Phew," and they shoot this harpoon out. That in some cases is short. This one's pretty doggone long, and it will inject poison into the surface. So this is how they capture prey. Something basically bumps into them. It also is pretty good defensively. You may know that clownfish have a coating on them that doesn't set these things off. That's why clownfish can live in a sea anemone and not get stung. Polyp versus medusa form. Basically, tentacles on the bottom, tentacles on the top. Tentacles on the bottom, medusa form, tentacles on the top, polyp form. And the mouth entrance is around the tentacles. Makes sense, you grab, put stuff in the mouth. Is that the polyp form has an attachment base. The medusa form has a swimmy bell that they pulse with in order to swim. Sorry. Different subgroups. This is more the coral section. These are the jellyfish. These are the sea anemones. Ctenophores may or may not be close relatives of these. My overheads, these types of overheads are fairly old. This connection is no longer as certain as it used to be. They're superficially similar to jellyfish. They don't have stinging cells though, and the genetics might indicate that maybe they shouldn't be lumped together as a sister group. It's another phylum, but there's a very strange way of putting this together. But you've got the polyp forms asexually budding off a medusa form, which does sexual reproduction and produces a small swimmy larva. Not going to swim very far. It's mostly going to drift down the bottom, find a good place to settle. But this is the alternation of generation cycle. Ordinarily these things would be on the bottom pointed up. Not entirely sure why they decided to do the picture this way. Is that, minus the labels, it really makes more sense to put it that way. But what the heck? And here's truly a dad joke. Dogbert demonstrates the arts of puns. Step 1, the setup. Tell me again about your uncle, the famous biologist. Uncle Albert won many awards for his work in breeding sea anemones. Sadly, he had little time for a social life. Step 2, the delivery from outside of swatting range. With anemones like that, who needs friends? Right, that's a pretty bad dad joke. And I'm a dad, so. Where do we stand? We'll get this group down. These are the flatworms. Flatworms, platyhelminthes, very fancy Latin word. Flatworms. It literally translates to flatworms. There are 3 major groups and some minor groups and some this is a sister group. Notice the way that these branch off. If the branches don't share, they're considered to be sister groups. And this one holds up relatively well. 2 of the groups are parasitic, and one of the groups is not. So, platy, like platter, helminthes. Helminthology is a broad study of worms. So people who are helminthologists often are experts in several different phyla. Worm shape is a very popular shape in the animal world. Animals that don't live inside other living things, don't live inside plants or animals that are not parasites, are called free living. I know it sounds like they're out getting coronavirus, but that's just the term that applies to them. So this includes flukes and tapeworms. We talked a little bit about this when we talked about apicomplexans, is that these go through two hosts for reproductive purposes. Sometimes they go through a bunch of other hosts just to get from one place to another. But they do asexual reproduction in the intermediate hosts. And for the flukes, it's virtually always a mollusk, and it's usually a snail. Could be a land snail, could be a water snail. And then the definitive host is where sexual reproduction happens. And flukes infect a whole bunch of different vertebrates and other types of animals. Tapeworms use a lot of different animals as intermediate hosts, including people. They're much more dangerous if you're an intermediate host because you wind up with the larvae embedded in your tissues, sometimes growing there quite large. And the tapeworms for the definitive host live inside the digestive system, where they live off of the food. Their outsides are covered with microvilli, so their outsides are built like the lining of an intestine. And a lot of times, they don't, they do release some chemicals that your body might respond to. They do release some chemicals that your immune system might respond to. But typically, having a tapeworm in your digestive system is not a huge health issue. Having a tapeworm larva where you're the intermediate host, that could be a big deal. There's a number of different flukes that are fairly nasty human diseases. Close to the top of the World Health Organization list of diseases to be eradicated is schistosomiasis, which is a fluke that it lives in the linings of the bladder for one species or the intestines for another species, and in order to get its offspring out of the body, it produces eggs with these spines on them. And so the eggs kind of roll out of the female up against the inside of the lining and kind of rip their way free to get out into the space of the intestine or the space of the bladder. But they also often get picked up and carried off into the general circulation, where tumbling through your little blood vessels is really not a great thing. And they can cause widespread damage and death. So they're a pretty nasty thing. Now the big deal of this group, no longer a jelly layer, an honest to goodness cell layer in the middle. And from that, organs and organ systems for sure, complexity. Some things are limited. These guys have gone on, okay, we're gonna get to be bigger animals, but you run into a problem with a multicellular system where you need to get oxygen to all the cells and you need to get waste materials away from all the cells. And if the cells are too far from a surface, the oxygen isn't gonna reach them. And the waste materials are gonna accumulate on their way out and do, so there's bad stuff in both directions. One of the ways around that is to have all of your cells very close to a surface. And if you're very flat, even the cells in the middle of a flatworm are pretty close to an oxygen acquiring surface and a waste removal surface. And, but it's obviously got limitations. What you'll see later are distribution systems, systems that can pick up the oxygen and carry it deeper into the body, can pick up waste materials and carry it back out to a surface. But mesoderm, these guys get very complex compared to cnidaria. If we had time, I would tell you the story about how I, as a freshman research student, I got outsmarted by these guys, but I really don't have time to do that. But, well, let me point with my little pointer. Pretty complex digestive system, pretty complex nervous system, not quite brains yet, but definitely cephalization. The opening of the digestive system isn't at the front end, it's in the middle of the body. Reproductive system, it's not unusual for platyhelminths to be male and female at the same time. Some of them are male and some of them are dioecious, some of them are monoecious. And they do actually, in some cases, have a waste removal system that involves a little tuft of flagella that moves fluid. And it looks supposed to look like a little candle. I've looked at a bunch of these guys. I've never seen one of these things actually look like a candle, but that's what they used to call them, flame cells. Okay, that's a few of the phyla.